Hi everyone, this is the first video on a series on phylogenetics, and today we'll be looking at phylogenetics and its place in taxonomy. With phylogenetics, we're not just able to classify animals and plants, but ourselves in the world in which we belong. The first thing I want to talk about is Linnaean Taxonomy. This is the most modern paradigm in which animals and all living organisms are classified. Most of us have learnt about this in school. It's a hierarchy system in which each level represents a consortium of organisms. It generally consists of domain, kingdom, phylum, class, order, family, genus, and species. The higher up you move in the hierarchy, the more representatives of species are present in that group. Whereas the more you go down, the more specific that group of organisms become until you have an individual species. This is all good and well, but how are living organisms classified according to this system? The general method still widely used today is the use of observations based on physical characteristics of animals and plants. For example, cat species have retractable nails and dogs don't. This is why we classify them as two distinct groups. There is one major flaw in this system, and that's a biased opinion based on your perspective of the physical characteristics you wish to use in your classification. For example, in a scenario where a person has to classify cats and dogs on the tree of life without ever having seen either of these two groups, how would they do it? One person might notice that cats are very similar in the way they look and they would thereby classify them very closely together. When they look at dogs, they might observe that there's a great variety among them and therefore classify them more diversely than they would cats. The problem with this, of course, is that we know that there are various cat species. However, dogs all belong to one species with various breeds. Think about it. In this scenario, you're not an expert on cats. Therefore, the only difference between a panther and a leopard might be the patterns on their fur. But when you look at dogs, you've got a Chihuahua and you've got a Great Dane. Of course you won't think they're the same species, and that's the problem with this method of taxonomy and classification. A fantastic example of the history of this sort of classification comes from the book Moby Dick by Herman Melville. There was an entire chapter dedicated to cetology, which is the study of whales. And in this chapter, there was a great debate on whether or not whales is a fish or a mammal. Today it's quite obvious. We know that whales are mammals. But back in the day, why could you blame them? They live in water, they have fins, they have flukes, they all look similar to fish. Why wouldn't you try to classify them as fish? And it was part of the fisheries. As this book highlights, it's not just the distinction between fish and whales, but between whale species themselves. There was quite a debate on which whales are more similar than other whales. How do we group them? And that was a debate even way back then. In the book, Herman Melville classified these whales according to his own perspective. But of course, based with modern classifications, they're not at all comparable. When using this method of classification, it's important to remember that observations are biased based on perspective. And here enters phylogenetics. This is an unbiased approach which eliminates observation to classify living organisms. This method also takes into account DNA-borne characteristics which cannot necessarily be distinguished between two organisms by observation alone, such as metabolic functions. Even though two organisms seem very similar when you look at the physical characteristics, they might have differences based on a metabolic level. A prime example of this is bacteria. Two bacterial cells look very similar, but their differences is in the metabolic capabilities. Phylogenetics works based on the comparison between similarities and differences in DNA sequences unique to different organisms. This does not only tell you which organisms are related to which, but how distantly related they are. This adds an extra dimension to classification of organisms. Basically, it all boils down to evolution. A single DNA sequence belonging to an individual organism will undergo change. These changes are very small and usually unnoticeable. However, after about 30,000 generations or so, these changes in the DNA become visible in physical characteristics, leading to the formation of new species. The severity of genetic changes between organisms indicate how similar or how distantly related they are, but all of it can be linked back to a common ancestor. A prime example of how the traditional classification using observations of physical characteristics can be misinterpreted is by the manatee or the dugong. Both of these animals can be found in the ocean, and they sort of look like sea cows. Both of these animals have characteristics unique to dolphins and whales, such as flukes and the ability to live in the ocean underwater, and also being a mammal. Therefore, it would be quite obvious to group them together. At least you would think that they are closely related to dolphins or whales, which is an obvious deduction to make. However, by using DNA sequencing and phylogenetics, scientists were able to determine that they are more closely related to elephants than they are to whales and dolphins. Now, there are some scientific disciplines out there that still classify organisms based on observation alone. In many cases, since most organisms have now been classified with DNA, they actually do correspond with their original classifications based on observation. But like I said, many still use observation alone, and this could lead to misinterpretations on their actual true position on the tree of life. From my own experience, organisms such as isopod, a parasite found in fish, is still classified based on measurements and microscopy. This is done by measuring the distance between two legs or the distance of the leg itself to determine the identity of that organism. Another discipline in which this still happens is the classification of diatoms, which is a type of microorganism, but they have a very unique cellular structure. It is this unique structure which allows them to be classified based on observation. However, as with bacteria, two organisms can be very similar but have different metabolic characteristics, which could lead them to be classified as two different species. Now, before everyone gets mad, I do understand why this is still being done. DNA extractions in these types of organisms can be very difficult, and it's therefore easier and more financially feasible to do them based on observations. The shape of a bacterial cell can look similar, if not identical, with hundreds of, if not thousands of different species. So how would one go about identifying the organisms? How would one distinguish between two individual species? Back in the old days, until quite recently actually, they would take these organisms and test their metabolic capabilities. A certain combination of metabolic capabilities would correspond to a specific organism, and that's how you would identify bacteria. These tests were extremely inaccurate, since one bacterial strain, which is thought to have the ability to perform a certain metabolic function, could not for that test, and therefore would be classified as something completely different, which is dangerous in the world of microbiology. For example, E. coli that naturally occurs in our digestive system is a known potential pathogen. Now, E. coli is not known to metabolize citrate. However, there have been strains found to metabolize citrate, and therefore they've been classified as a different organism. I don't think I have to tell you, but to take a potential pathogen and classify it as a non-harmful bacteria could have some dire consequences, especially in food microbiology. Phylogenetics have allowed us to use DNA to classify these organisms and not at all their physical or metabolic characteristics. It's important to remember that only 1% of all estimated known bacteria have been grown in a laboratory. The other 99%, we have no idea what they look like or where they sit on the tree of life. Luckily, advancements in DNA sequencing have allowed us to analyze millions of DNA sequences simultaneously. Using this DNA sequencing technology, we are able to classify thousands of bacteria by targeting key DNA sequences unique to those organisms. Even though these bacteria have never been seen before, we can use an inverted approach to predict their environmental niche. We can, for example, take an unknown organism, and then the most closely related known species can be used as a reference point. With this reference, we can estimate the type of metabolic functions and also the environment in which this organism will thrive and survive. Taxonomy is important so that we can find our place on the tree of life. Now, with the use of DNA and phylogenetics, it allowed us not only to determine how we are related to each other, but also how we have been geographically distributed over history. Other deductions can also be made about how we migrated throughout the globe, forming new cultures and civilizations. Not only does this reveal our anthropological history, but it also can be used to predict the course of our future. From here, we will look at the practical applications of DNA analysis for phylogenetics. I hope you follow this series and the future videos so that you can broaden your knowledge and enhance your skill set. Stay safe, stay smart, and thank you for watching.